Hey, let's get going tonight, huh? Thank you for being here to the Muskegon Community College Lecture Series. I'm happy to have our speaker, Dr. Emily Dolson, who will be speaking on building lifelike ecology and evolution in a computer, which you can read. So looking forward to her talk. The lecture series has been going on for well over 20 years at MCC. And I've had a lot of prestigious speakers along the years, and it will continue. This is our last one of this semester, but it will continue next semester. So get your pens out. If you need to borrow one, here's one. No, I'm kidding. Well, you got one. Thank you. First off will be on January 29th. It's our next installment of Pecha Kucha, which are 20 slides, 20 seconds per slide. And Dr. Kilborn will be gracing our stage and giving a great presentation. He's a little hesitant, so now I have confirmed his presence. But anyways, it should be a good we'll probably have about 10 speakers. If you don't like one, they only last 6 minutes, 40 seconds each, so you might like the second one. But last year we did it was great fun and, a good winter activity. The second one we're doing is on February 13th, called Fact and Fiction, Media Consumption and History in America. This is part of Black History Month, and it's a panel discussion about the media and Marion Stokes, who recorded media full-time for over 30 years and was quite an interesting person. And there's a documentary about her called The Recorder that we'll be showing part of, and then have a panel discussion about it. And that'll be in Overbrook Theater. Both of those are actually be in Overbrook Theater, so hope to see you there. Tonight's lecture will last for about an hour or so, and then there'll be time for Q&A afterwards. So as you're listening along, get your questions ready, and Dr. Dolson will, I'm sure, be happy to answer all questions that you have. Now a little bit about Dr. Dolson. Undergrad, Swarthmore College in Pennsylvania. With a dual major in computer science and biology, which seems to be still what she's doing, is both of those things. And so, and then did her PhD at Michigan State University, where she currently teaches. She runs the Evolutionary Control and Digital Ecologies Lab, and is assistant professor at Michigan State in the departments of computer science and engineering and ecology, evolution, and behavior, and the Beacon Center for Evolution in Action. She was previously a postdoc in the theory division at the Cleveland Clinic Lerner Research Institute, where she studied the eco-evolutionary dynamics in cancer. She has also been the chair of the International Society for Artificial Life Student Group, interesting, and she published a lot of papers. I'm not going to go through them all, but I picked one out that I thought was interesting, and maybe here's a word that some of my students may have heard of: the prisoner's dilemma, memory, and the early evolution of intelligence. She did her PhD in the Digital Evolution Lab at Michigan State, where she's come back home to teach. In her spare time, she enjoys playing board games and the tin whistle, which I don't know if you brought that with you.
I didn't.
We have any music people here that may get one? Oh, anyways, I'd love to hear that. All right. Well, anyways, please help me give a warm welcome to Dr. Emily Dolson.
Hi, everyone. Thank you so much for coming. I'm really excited to be able to talk about all of this stuff. So my research is really interdisciplinary, and this talk is kind of set up as like a tour through kind of the how and the why of building lifelike ecology and evolution in a computer. So I'm going to kind of illustrate those ideas with examples from my research and other people's research that I think is cool. As Andrew said, there will be time for questions at the end, but if you have any burning questions partway through, feel free to interrupt me and ask. I want to talk about whatever is most interesting to you. I could go into more detail about any of these things. I'm happy to focus wherever you guys are most interested. But so let's get started with the why side of this. So why do I want to build lifelike evolution and ecology in a computer? Well, the reason is I am really passionate about answering this question. How can we steer evolving ecological communities towards target states? Okay, and that's really abstract. So let's think about some examples of situations where it would be really great to do that. So one that I worked on during my postdoc and have been continuing to work on since then is cancer. So cancer is a population and it evolves on a time scale that is relevant to a human that has cancer. It actually will often evolve resistance to drugs and other therapies that we use to treat it, which is obviously a really big problem. A lot of you are probably familiar with the fact that we keep inventing really great new drugs that can help treat cancer, but we still haven't cured it. And a big part of the reason for that is that the cancer evolves resistance. Cancer is also an ecological community. Within a tumor, you'll have multiple types of cells that are interacting with each other. And often the way resistance evolves actually has to do with those interactions. Like perhaps one cell will produce a compound that gives resistance to it and all of its neighboring cells. And so understanding those interactions is really important for predicting what's going to happen. And so obviously the target state that we would like to steer the evolution of this community towards is one where it's dead, basically, or at least where we can keep it under control at a very low level. So dead is a nice simple target state. We might want to steer things towards more complex states as well. So the gut microbiome is another example. We've got this really complex collection of bacteria and viruses and fungi and other things in our guts. And more we're realizing that the set of what exactly we've got in our guts is really important for our well-being, and it has health implications. We don't totally know yet what exact composition of microbes we want in our guts, but we know that it matters. And so my hope is that by the time me and other people asking this ecoevolutionary question kind of have our answers about how we steer evolution, maybe the gut microbiome researchers will have figured out what state we actually want to steer our gut microbiome to. But yeah, clearly some states would be better than others, and it's definitely evolving over the course of our lives because all of these organisms have really short generations, and so that gives them time to evolve. Another really different instance of this problem that I also work on is evolutionary computation. So this is a machine learning algorithm that basically works based on evolutionary principles. So we'll create a population of things that are potentially solutions to some sort of computational problem. So here I've got an equation. It's represented in this tree form, but it's turns out to just be this equation here. And it's maybe trying to be a map from like input to output. Like it's trying to figure out the best way to explain like why we have a set of inputs and outputs. This could be any computational problem. This is just an example. And so if we're using an evolutionary algorithm to solve this problem, what we would do is we would evaluate all of our possible solutions. Initially, they'd all be really bad. We'd give this equation the inputs and it wouldn't give us anything at all like the outputs that we want. But some would be better than others. And so we would choose the ones that were closer to solving the problem than others, and we'd make copies of them, and we'd make slight mutations to those copies. And then over time, for the same reason that evolution works in nature, we would evolve equations that are better at solving our problem. And it doesn't have to be equations. This could be anything. I'll talk about some more examples later. This turns out to also be an ecological community for reasons I'll talk about later. But basically, the way we set up these evolutionary algorithms ends up creating interactions between the solutions in our population. So in this case, the target state is we would like to guide the population to a state where it has solved our problem. So it's a very different state than, for instance, the cancer. It's actually kind of the opposite. We want to help this population evolve complex traits and solutions. Whereas in cancer, usually if that's happening, it means something bad is happening. It's evolving, resistance or something. And so, working on both of these problems is fun because I can take the things that make my job harder on one of them and apply that to helping solve the other one. And then lastly, just one other example. So this is the one that, we're nowhere near being able to solve this, but it's kind of what keeps me up at night and is, my ultimate motivation. is kind of larger scale ecosystems that we have on Earth, like agricultural ecosystems. So when we grow food, obviously we have the crops, but we also have this whole ecosystem around them. We have soil microbes, we have pollinators, we have all kinds of other interacting parts that are helping those crops grow well. And so as our climate changes, probably some members of that ecosystem are going to struggle in our new climate. And so my hope, again, is that if we solve this problem, soon enough, by the time this is starting to affect agricultural ecosystems, we can maybe kind of guide the evolution of the members of those ecosystems to a place where the ecosystem still functions. We can still grow our food. Maybe we have different soil microbes. Maybe our soil microbes have evolved to be heat resistant or moisture resistant or whatever the problem is. But we still have plants that we can eat. Okay, so that's kind of like the big picture, set of examples of why I'm interested in this question. I want to also just briefly talk about what I'm actually personally working on within this space. So I kind of see my lab's research as this diagram here. So I have this kind of central question about, really abstract ecoevolutionary theory. How do we expect, populations in general to change over time when there's ecology and evolution happening? But that's really abstract. That's hard to think about and hard to work on. So to keep myself anchored in, actual problems that matter, I basically have a bunch of areas of application that I work on, and I try in each area of application to think about, okay, what is the next thing that we need to understand about this system to be able to control evolution in it? And then whenever I do that, I learn things about ecoevolutionary theory in general that then I can apply back out to all the other things. Because really, these things are going to sound different when I say them, but they're all fundamentally the same problem. It's the same process. So yeah, stuff I currently have going on in my lab is I have a research direction on endosymbiosis. So that's basically, one species living inside another. So that could be parasitism, where the species living in the other is hurting it. It could be mutualism, where the species living in the other is helping it. It could be something in between. We have a lot of questions about how those relationships evolve. And so this is work I'm doing in collaboration with Anya Vostenaar at Carleton College. And the goal is that this can eventually kind of inform gut microbiome research, but we're not there yet. The gut microbiome is very complicated. We're just beginning to tease apart how these interactions work. I've also got an evolutionary medicine direction, so that includes the cancer stuff that I talked about. The same exact thing that happens in cancer happens in antibiotic-resistant bacteria, or malaria that's resistant to antimalarials. Really pick any kind of thing that makes you sick that is a population, and you'll have this problem of evolving resistance. And so I do all of that in collaboration with Jake Scott at Cleveland Clinic, who is my postdoc advisor. A new direction I just picked up that we haven't really started yet because we just got the grant for this is more of an evolutionary epidemiology direction. So epidemiology is the study of how disease spreads within a population. Been a lot of really important epidemiology work lately to figure out how can we stop people from getting the flu? How can we stop people from getting COVID? But historically, it's been pretty hard to factor in evolution and think about what are going to be the evolutionary impacts of a given vaccine program or a given antiviral program. And so the reason that there hasn't been much work on that is actually computational limitations. And so part of what we're doing here is actually building much faster models, and there's a whole computer science algorithm development thing going on under the hood there that I'm not going to talk about unless people are curious. But the goal is to kind of use that as a test case to try and do some actual evolutionary epidemiology where we have, our goal is to make a national model of COVID evolution and see if we can predict what the impacts of different interventions would have been. We'll see if we make it to national level. Maybe we'll do state level first. But we think we can dramatically scale it up. I've also got this origin of life research direction that, again, I'm not going to talk about much, but feel free to ask questions. It's really kind of fascinating and mind-bending. Basically, there's this idea that my collaborator, David Baum at University of Wisconsin-Madison, I should have mentioned, my evolutionary epidemiology collaborators are Matthew Moreno and Luis Simon at University of Michigan. Right, so David Baum at University of Wisconsin-Madison is my origin of life collaborator. He has a wet lab where he actually does chemical evolution experiments. So he has like little vials of different chemicals and he'll like propagate them over time and see if he can get things kind of like evolution emerging out of them. And so our whole question with that is basically, the origin of life is kind of in some sense the origin of evolution. And so we're trying to figure out like what factors lead to that. And yeah, feel free to ask me more about that. It's its whole own topic, so I'm gonna keep going in the interest of time. And then the other topic I work on in my lab, because it's just such a great model system for studying all of these other questions, is evolutionary computation, which I mentioned before. So that, one of my main collaborators is Alex Lalgini at Grand Valley State University. So I've got lots of great, Michigan collaborations going on. It's a good place to be for this kind of research. Okay, so these are the questions I'm interested in, but how do we attack these questions? Well, it's actually pretty hard because evolution takes a long time. I don't know if any of you are aware of Rich Lenski's long-term evolution experiment. It was at Michigan State University for a long time. It's actually at University of Texas right now, but it might come back. And so he's been evolving E. coli for I think 35 years now. It's a little bit older than I am, this experiment. And in that time, they've gotten through around 80,000 generations. Which is amazing. Like, this is a fantastic experiment. We've learned so much from it, but it's taken 35 years. We can do wet lab evolution experiments that are faster than that, but there's still always a pretty substantial time cost because evolution takes time. That's part of how it works. So, we can't necessarily do wet lab experiments for every evolutionary question that we might have. Another challenge to studying this in the lab or the field is it's hard to measure all the things we care about. I said that these interactions between species, this ecology, is important, but measuring those interactions is actually really hard. And so it's hard to build a theoretical understanding of something if we don't even know that we are like measuring the thing that we think we are. And then also it's really hard to do a lot of the experimental controls that we would ideally like to do in the lab and the field. A factor that's incredibly important to kind of the outcome of evolution is the mutation rate. Like, how likely is it that a given spot in your genome is going to change? And we can't just like reach into a real world organism and change its mutation rate. That's not how biology works or how physics works. And so there's a lot of limitations. And so for this reason, I think to answer this question, we really need a multi-pronged attack. We need to use a lot of different methods together. So a lot of you might be familiar with the idea that if we really want to understand how something works in the real world, we should go and look at it in the real world. We should do field research. Any abstraction beyond that is potentially going to risk missing something that happens in the real world that matters for that exact thing. However, there's a lot of things we just can't do in the field. Some of them are unethical. Some of them are just impossible. Some of them are impractical. And so for that reason, we do a lot of work in the lab. We have to. It's the only way to answer certain questions. Stepping back towards computational systems like simulations and artificial life, which I'll explain what that is momentarily, is kind of the same trade-off. We're again sacrificing some of the naturalness of the dynamics, but we're gaining more ability to do experiments fast, with the controls we want, and to perfectly collect the data that we want. And we can step even farther back to pure mathematical models. And if we have a mathematical model of something that's accurate, that's obviously very easy to perfectly understand because it's literally an equation. Like, that is the answer for how something works. But there's also a long history of mathematical models making abstractions that turn out to be misleading and give us the wrong answer. There was a long period of time where people were confused that cooperation could evolve because it seemed like it shouldn't be possible based on mathematical models. And it turns out that it's because the mathematical models were assuming that everything interacts with everything, which isn't how things work in the real world. You know, things interact with things that are close to them in the real world, and that turns out to really matter for whether we can evolve cooperation. So anytime, you know, we're scaling back, we're potentially missing things like that. We're potentially making abstractions that reduce our ability to understand what's going to happen. And so, yeah, that's why I feel like we need all of these. They're all important. So I focus specifically on the simulations and artificial life side because that's my skill set. But I make sure to collaborate with people who do all of these others because I don't think we're going to get answers to these important questions unless we work together like that. Okay, so that finally brings me to the how of building ecology and evolution in a computer. So simulations and artificial life are both instances of doing that. And fortunately, evolution is easier to build in a computer than you might think. And that's because evolution kind of has three fundamental ingredients that it needs to work. So it needs inheritance. So you need self-replicating organisms where the offspring are similar to their parents. So they, you know, inherit their genetic material. We're pretty comfortable with that as biological species. You know, our kids tend to look like their parents do. And so we need inheritance. We also need differential reproductive success. What that means is that some things need to have more offspring than other things, basically. And the reasons that they have more offspring need to be heritable. So they need to be part of that stuff that they're inheriting from their parents. And then we need some source of variation. The population can't be all the same, or we're not going to get any change. So that's often mutations in biology, but it could be other things. And so the really cool thing about evolution is that if we have these three things, it's kind of a mathematical fact that it's going to happen. There's no real way around it. The things that make more of themselves are going to be more plentiful. And so that's the crux of evolution by natural selection. I actually want to just pause here. Are there any questions about this idea before I move on? Does that feel comfy in everyone's head? Cool, okay. So the upshot of these being kind of the three critical ingredients for evolution is that we can put them in a computer and then we can have evolution happen in a computer. And there's kind of two ways we can do this. There's a continuum of ways we can do this. There are two extremes to that continuum. So we can assemble them in very targeted ways to make simulations of really specific evolutionary scenarios. And so I'm going to start by talking about an example of that, and then we will talk about the other one a bit later. So for example, I said antibiotic resistance is the kind of thing that this research can help inform us solving. If we wanted to build a model of antibiotic resistance evolution in a computer, we could start out with something like this. And we can say, okay, we know that for some bacteria that we care about and some antibiotic that we care about, there are 4 genes that bacteria is likely to evolve that will give it resistance. And so if we want to simplify that, we can just say, okay, each of these bacteria are represented by the set of genes that they have and don't have. So here the zeros are indicating that they don't have the gene, the ones are indicating that they do. So this example genotype would have the second resistance gene and none of the others. And so if we, imagine this world, we can measure the fitness or the reproduction rate of bacteria with each of these combinations of genes. And if we want this model to be, directly modeling something in the real world, we can do this measurement in the real world. And if we get this, ta-da, we have differential reproductive success. Because we have some things making more copies of themselves than others. These genes are also heritable. The offspring are going to have the same genes as their parents, so we have heritability. And we can allow mutations. We can say that offspring can be born with one gene being different from their parent. So for example, this one could have an offspring that has this third gene as well. And so we would get a mutation to kind of an adjacent genotype. And so that gives us variation. So we have our 3 ingredients for evolution. If we put this into a model, we're going to get evolution. And it's going to be evolution in this really constrained space that's based on this exact model of this bacteria and this antibiotic, we can model what will happen, in that actual real-world scenario. And so, yeah, as an example of what we could do with this, I'm going to talk briefly about some work that's been done by Ishan King. He's an MD PhD student at Cleveland Clinic, or actually Case Western University. They are integrated closely with Cleveland Clinic. And so I mentored him on this during my postdoc, and he is probably just about to finally submit it to a journal, which is super exciting. I just got a final draft from him. And so he's actually done some really cool work that combines the wet lab and the computational side. So he literally measured all of these things for, I believe it was E. coli evolving resistance to an antibiotic called ampicillin. That doesn't matter for the purposes of what I'm going to talk about, but just so you know, these are actually based on things we measured in the lab. So we can take all that information that I just said, the differential growth rates and the mutations, and turn it into this concept called a fitness landscape. So a fitness landscape is basically a space where we expect evolution to play out. So the way to read this is each of these circles here is a genotype. So this one with all the zeros has none of the resistance genes. This one with one has, the last resistance gene, and so on. So we have all 16 possible combinations of having different resistance genes. The lines connecting them indicate which ones are mutationally adjacent. So basically, which ones only involve gaining or losing a single resistance gene. And then the colors of these circles indicate the fitness of each of these genotypes. So what is their growth rate going to be? And so if we kind of take this all together, this will be kind of a prediction for where we expect evolution to go. We generally expect it to kind of climb gradients in this space. If we start out with no resistance genes and we're adjacent to this genotype that's much fitter, we would probably expect eventually we're going to get a mutation that gives us this genotype, and it's going to be a lot fitter, and so suddenly we're going to have a lot of them. And so that's something we could do with one fitness landscape. However, I've glossed over a really important detail, which is that when you measure these fitnesses, you need to measure them in the context of a specific environment. And so the environment that's really relevant here is whether the antibiotic is present. So this is the fitness landscape for kind of a medium amount of the antibiotic. But we're going to get really different fitness landscapes if we have different amounts of it. So if we don't have that much antibiotic, having all the resistance genes is actually not that great. The fitness is pretty low, probably because there's some cost to having them if they're not protecting you from an antibiotic. Whereas as we increase the concentration, like up to high, here it's actually really good to have all of the resistance genes. And so we expect different things to happen at different concentrations. Using this information, we can actually model even what we would expect to have happen if the concentration changes over time. So this is a slightly different fitness landscape, but it's the same idea. So here we're starting with the population all having no resistance, but we're going to gradually increase the concentration soon. Yeah, there it goes. And then as the concentration increases, you can see it becomes advantageous to go into the rest of the landscape. And eventually we're going to end up evolving the version that has all of the resistance genes. So this is an example of the kind of sequence of concentrations you would probably not want to subject your population to because it's resulting in antibiotic resistance. That's bad. But the point is we can model this and we can predict what the outcome of giving a different sequence of drug concentrations is going to be so that maybe we could avoid doing this. And so the fact that we can model evolution in response to changing amounts of a drug is actually really important because we don't always think about this, but when you take like an antibiotic pill, it's not like suddenly everywhere in your body the antibiotic is at the specific level that your doctor wants it at. It takes time for your body to break it down, metabolize it, for it to go into your blood or into wherever else it needs to be to treat your infection. And so in reality, when you take antibiotics or other drugs, they kind of follow this curve where like they'll gradually increase, they'll hit a peak, they'll start dropping off. Maybe at that point you would take another dose, which would push you up higher. And so it can actually take a lot of doses for you to reach maybe the stable amount that your doctor would want it at. And at all points, the concentration in your body is constantly changing. This is actually a thing that's kind of been overlooked by some past research, so that's why we especially wanted to study it here. So using this model, we can then ask some really important and practically relevant questions, like, how bad is it if you miss a dose of your antibiotic? I'm sure a lot of us have had antibiotics prescribed to us by doctors that we have to take on a certain schedule, and it's easy to forget or to take a dose late. How big of a problem is that? Well, I want to again preface this by saying this is this specific bacteria in this specific drug. Don't try and assume that this will apply to other bacteria or other drugs. The point is, for any given situation like this, we could use a model like this to figure out what the risk of evolving resistance under different circumstances is. So here, we've got the concentrations we'd expect over time if you took the drugs as you were supposed to. We can then also model trajectories of the amount of drug in your system if you forgot with various probabilities. So these are just examples. So like here, you have a 20% chance of forgetting to take a dose. Obviously, there's a lot of different trajectories you could actually get because you could forget different doses. This is just an example of what that might look like. And then here's if you forget to take 40% of your doses, and here's 60%, and here's 80%. And so you can see, if you forget too many, not only do you not have very much time with the drug in your system, you also potentially don't reach this actual concentration that you were aiming to reach, which in some cases could be a problem. So then what we can do with these simulated courses of concentrations is we can predict how the drug would evolve under these, how the bacteria would evolve when given these different drugs. And so we made 100 different possible courses that had each of these probabilities of forgetting. And this is what we ended up with. So this is the chance of treatment success over time. You can see in this particular case, if you don't forget any of your doses, you're pretty much guaranteed that it's going to work. And so by work, I mean all the bacteria die. If you forget some, there is a chance that they're not all going to die. And generally when this happens, it's because some of them have evolved resistance. And so, again, limited context, limited scope. Don't overinterpret this. But this kind of speaks to why it's important to take all of your doses of your antibiotic, because you don't want to evolve resistant bacteria in your body. So this is important, for helping, explain why you need to take your antibiotics. It's also useful though, because people do forget, and if we understand kind of what state the population is likely to be in when that happens, we can better respond. We can better figure out, okay, is there a different antibiotic we should give next to treat this, population that's now resistant to the first one? Or, how can we still help you even if you maybe ended up in this unfortunate situation? Okay, so that's my example of a simple evolutionary simulation. Any questions about that before I keep going? Oh, yeah, question?
I'm really bothered that they all go to 50%. So what if you put in 100% chance that you missed your dose? Does it still go to 50% and you get better in 12 days?
Oh, good question.
If you take 2 doses, it says it's the same as taking 6, right? Or 60%.
That is a really good question. Yeah, I'm going to ask you, Sean, that. So I know we've also done some work on which doses are most important. And so it turns out it's a lot more important to take the early doses than the late doses. Right. And so, this is the average across a bunch of different courses where you forgot 80%. So I'm not sure if this is being driven by the fact that sometimes you take it early and that knocks down the population enough that it doesn't evolve resistance. If you don't give them any of the drug, they probably won't evolve resistance to it, but also you'll still have a bunch of bacteria in your body, which may be a problem. Great question. Are there any other questions? Cool. Okay, so that's a nice, simple system. I should check I'm doing on time. Okay, I will speed up a little bit. So this is an example of how we can make a really targeted evolutionary model in a computer. So this is great if we have a specific question like this that we want to answer. But it's not so great if we want to learn something really general about how evolution works. And so kind of the other side of the coin on creating evolution in a computer is we can make these really rich, complex, open-ended evolutionary systems. And so these are mostly found in the field of artificial life, which is the study of life as it could be. And basically, it's kind of a playground of like fun, different systems. I'm going to talk about the software systems, but there are also people who have wet lab and robotic systems. Basically, all of them are kind of, built on these same lifelike principles of things like evolution and ecology, but they all have, just different twists to how exactly they implement those ingredients. And so they all behave really differently, and we can learn different things from all of them. And none of them are going to act the same way as evolution does in nature, but sometimes that's actually a good thing. Because if we want to learn something that's generally true across all evolving systems, seeing the same behavior in one of these really different systems that we see in nature is a really good indication that any other system that works on the principles of evolution would also act that way. So this has implications for astrobiology, what we might expect to see if there were life on other planets. It also just has implications for, learning fundamental rules about evolution instead of rules that are specific to evolution in DNA. Because every system, including DNA like we have, is going to have its quirks. Obviously, it's important for us to understand the quirks of DNA-based evolution because that's what we're dealing with most of the time. But it's also useful to understand how things might be if we were, encoded differently. Okay, so I wonder how this video is going to work. Okay, I think I can just use my mouse. That'll work. Okay, so I want to give you a flavor of what these artificial life systems are like. So I'm going to talk about one of the classic ones, Evita. So this is basically a system of self-replicating computer code. So these organisms are kind of like domesticated computer viruses. Their genomes are literally a sequence of really, really simple computer instructions, and they spend their lives just executing those instructions and doing whatever the result is. So here's an animation. So here, the first instruction in this one's genome is input. So it's going to take a number from the environment, because that's what the input instruction does, and it's just going to store it somewhere in its memory. It's going to go on to its next instruction, which happens to be a pop instruction. That happens to take something from one part of its memory and put it somewhere else. That's an instruction that doesn't do anything. It's going to run this add instruction, and it's going to add those two numbers together and store the result somewhere. And it's going to run an output instruction, which will spit that number back into the environment. And here, the environment might notice, oh, this organism took in two numbers and outputted the result of adding them. It did addition. So it's going to keep going. It's going to keep executing its genome until eventually it is going to hit an instruction that tells it to allocate space somewhere else in the world. And then it's going to go through a loop where it is executing a sequence of instructions that tell it to copy the instruction in one part of its genome into the new space in the world. And so it's literally just looping over its genome and copying each instruction into this new spot until it's done and it's made a copy of itself. This is going to keep happening. Sometimes there will be mutations during that copying process. And this will keep spreading out until it fills the world. Whenever an organism copies itself into a new cell, if there is something already in that cell, that thing is going to get overridden. So there's pressure to copy yourself fast before you get stepped on, basically. And so that creates differential reproductive success because some things are copying themselves faster, and so they're going to make more copies before something copies over them. So we have differential reproductive success. We have heritability because they're literally copying each instruction in their genome into a new place in the world. And we have variation because they have those mutations. And so again, we have the three ingredients for evolution by natural selection, and we get it. So in the simple case, they evolve to copy themselves more and more efficiently. In the complex case, we'll sometimes reward them for doing tasks like that addition task, so they can evolve to do different computations, basically. So for those of you who maybe haven't written any code before, that was pretty fast. I don't know, are there questions? Anything that can help clarify? Yeah?
So when it copies over into a new cell and gets overridden, does that mean the one that overrode it is more stronger in a way?
It could mean that. So statistically, it'll definitely be true. In any individual case, it could just be that the one that got copied over got unlucky. Like maybe it was super fast at copying itself and something really slow just happened to copy over it. That could totally happen. But in general, the faster ones will make more copies of themselves before they get overwritten. And that's true of biology too. Like, plenty of things can be, theoretically really fit. Like they have all the right traits and, they just get struck by lightning. Cool. Any other questions? Sorry. Cool. Okay, so Avita is a classic example of an artificial life system. Does it not want to advance now? There we go. Okay, and so as an example of the kind of research we can do in this system, we can ask much more abstract questions about how evolution happens. So here, I was really interested in this question of how does spatial heterogeneity, or basically the fact that there are different things in different places of the world, how does that influence evolution? So if you think about things evolving in nature, there's some places where it's useful to be resistant to cold. There's other places where it's useful to be resistant to heat. There's some places where it's useful to be able to eat certain plants or different plants. And all these traits, are only useful in certain places. And so in Avita, I created a situation like that. So I basically put different resources in different parts of the world. What that means in practice is that different tasks, like that addition task or other really simple computational tasks, are rewarded in some parts of the world but not others. And so the color of the background here indicates basically kind of how many and what complexity of tasks are rewarded there. And then the circles in the foreground indicate what creatures, what computer programs ended up there. So the different concentric circles basically say which traits they have or which tasks they're able to solve. So like, maybe this green circle means they can do addition. So all the ones with the green circle can do addition. Some of the others can do other tasks as well. And what you can see is we end up with different things in different places, which is, kind of what you'd expect. It's what we see in nature. So that's a good starting point. That then lets us ask a deeper question that we can't ask as easily in nature, which is a thing that conservation biologists have wondered about for a long time. So there's places in the world where we see that there seems like there's more evolutionary diversity. Like it looks like there are more different species with different traits. And there's a question, is it just that a lot of different species could survive there? Or is the rate of evolution of new traits there actually higher? Like, is this a hot spot of evolution? Or is that not a thing? It's hard to do that experiment in real life because we only have one copy of the Earth. We can't do replications. But we can have as many copies of Avita as we want. So what I did was I created 8 different environments that had, kind of different resources in different places. And I ran 100 runs of Avita in each one. And I looked at what places in space each task evolved for the first time. And I did some math. And basically it turns out that we do see these hot spots of evolution. So there's some regions where it's more likely that a given trait will evolve for the first time. This doesn't tell us if, the Amazon rainforest is a hot spot of evolution. It doesn't even tell us if there are actually hot spots of evolution on Earth. But what it does tell us is that it's not crazy to think that if we have a system based on these principles, that there might be hot spots of evolution. So this is just one example of research that I've done with Avita. There's a lot of other research, but I also just want to show you some other artificial life systems because they're really fun. The one that I love is soft robots. So these are robots that are composed of kind of cubes of different materials. So the red material is muscle that expands and then contracts. There's another kind of muscle that does the opposite, that green muscle. The blue is kind of bone, it's rigid, and the cyan is kind of just soft tissue. And so you can create a population of these robots, and what they do is they, measure how fast each one of them can walk. They take the ones that walk the fastest, they make more of them, they mutate those. Over time, they evolve these soft robots that are able to walk faster. And you can see they get a really wide diversity of gaits. And we can see if we look over time, like kind of how this process plays out. So first we start with this cube that's just bouncing along a little bit. We get a bouncier cube. Eventually we get a mutation that kind of starts to give it legs almost. Get some more legs, etc. And so you can see how this is able to build up incrementally through mutations. Now we have some like real legs. We're starting to get the different muscle types playing a role here. And they're just really fun to watch too. So sock robotics are an example of, of evolutionary computation, which I already explained. So basically it's this cycle where we have a population of solutions, in that case, robots. We evaluate how good they are. In this case, how fast do they go? We select some to reproduce. Usually it's the fastest, sometimes it's not. I'll talk about that later if I have time. And then we apply mutations to their offspring, and we keep going. And we get faster and faster robots that have all these cool different ways of walking and existing. We can do a lot of other stuff with evolutionary computation too. One of the classic results is this antenna that was evolved for NASA. And you might be thinking, this looks really weird for an antenna. It does. That's actually one of the big strengths of evolutionary computation. It's really creative. It comes up with things that a human wouldn't. This antenna turns out to be really good because it's a lot lighter than a conventional antenna. And this was designed to go up on spaceships, so it really matters that it's light, because every ounce counts for sending things into space. More recently, I've actually been working with some folks at JPL to evolve more antennas. So this is one that they came up with to detect high-energy neutrinos. So this kind of antenna basically goes into the ice in Antarctica, and it listens for neutrinos arriving. And this one that we evolved is slightly better than the one that's currently in the ice, and we are working on evolving even better ones. Another just really cool example of evolutionary computation is this project where they evolved robots that can walk in different ways to adapt to damage. So this is a 6-legged robot, and so you can imagine one of its 6 legs might get damaged, and it would still want a way to walk. So what they did is they evolved a whole library of different gaits, so walking patterns, that used each leg a different percentage of the time. And so for example, here we have, so in this case, this leg is injured, and so it turns to a strategy for walking that doesn't require with that leg. The other thing I just love about this experiment is, so they were trying to evolve, all possible combinations of using and not using different legs. They didn't bother to tell it not to try to evolve a gait that doesn't use any of its legs. They didn't think they would actually get one, but then at the end they looked and there was a gait in the category for not using any of its legs. They were very curious how that worked, and so they looked at it, and this is what they found. Oh, whoops, no. There. It flips on its back, and it kind of shimmies on its shoulders. So again, these algorithms are very creative. They've also done other really cool stuff. I'm going to skip over this in the interest of time. Okay, so I've just talked a ton about evolution, but I said building ecology in these systems is also really important, and ecology is also important for, how these systems evolve. So how do we build ecology? Well, it's less straightforward than building evolution in some ways. There aren't ingredients for ecology in the same way that there are ingredients for evolution. But fundamentally, what ecology is either interactions between the different species. I have species in scare quotes there because species is a really loaded word. I don't usually use it, but it's the easiest thing to think about. So if you're a biologist that knows about biological species concepts, feel free to think about this as phenotypes or genotypes. But if species feels comfortable, you can think about them as species. Ecology could also be interactions between species and their environment. And so there's a ton of ways you could create these things. And in these artificial life systems, there are a ton of ways that we create these. So yeah, it's kind of an open sandbox. But there are kind of a few classic ways that we think about them. So one of these lenses is game theory. So I think I heard a reference to Prisoner's Dilemma earlier. So some of you are probably familiar with this idea. Yeah, is everyone familiar with Prisoner's Dilemma? Or Okay, I'm seeing nods. Yeah, okay, I'll go over this quickly. So yeah, basically, Prisoner's Dilemma is kind of a classic social game. The idea is you have two prisoners. Either of them could choose to cooperate, where they, don't tell the police what they know about each other, or defect, basically turn the other prisoner in. And there are different outcomes associated with each combination of choices. So if they both cooperate, they both get 1 year in prison. Not great, but not terrible. If one of them cooperates and one of them defects, the one that defected doesn't get any time in prison, but the one that did not gets a lot of time in prison. And then if both of them defect, they both get a lot of time in prison, although slightly less than the scenario where only one of them defected. So basically, this is a situation where theoretically the best global outcome would be for them to cooperate, but depending on the exact rules, it's often hard to get cooperation to be stable because, in any individual round of playing this, if they both cooperated, it would have been better for either one of them to have defected instead and let the other one cooperate. And so there's, mountains of research on this and on, what we should expect from this under all kinds of different circumstances. I'm not going to talk about any of that, but what I wanted to say is that this is a general lens that we can use to describe the kind of interactions that different species or different cells are having with each other. And we can actually abstract it a little bit. So what really matters is what the numbers in this payoff matrix are. And if we do some math, we can show that really what matters is this number and this number. And using that, we can actually kind of plot the space of two-player games of this form on this space here. And there's different regions that have qualitatively different games that, economists and stuff would say, we have different expectations from. This is a whole fascinating field that I'm not going to get into very much, other than to say that we actually use this a lot in cancer evolution to kind of classify how the cancer cells are interacting with each other and to try and make predictions about what course of treatment is going to make sense given the nature of those interactions. And I can talk way more about that if people are curious during questions. Right, so yeah, I've done some work on game theory in cancer. I'm running a little low on time, so I'm going to not talk about it. The other way that we talk about ecology in these systems is with species interaction networks. These are a lot more kind of open-ended and free-form. That also means they're harder to make generalizations about, so, pros and cons. The way you read these is the nodes in them each represent a species. Standard caveats I already gave about the word species. And then the edges indicate how they're interacting with each other. So a red edge here, or a negatively valued edge, indicates a harmful interaction. So A is hurting B. That means B's fitness is lower if A is present than if A is absent. These edges can have different intensities. So here A is hurting B a lot, B is hurting A a little bit. We can also have beneficial interactions. So here, B is helping C. So C's fitness is going to be higher if B is present than if B is absent. And so using this general framework, we can describe the interactions in any population. And so a place where I've used this a lot, actually, is in evolutionary computation. So I said before that when we select solutions to reproduce, we usually select the best ones, but not always. And the reason we don't always select the best ones is it's also really important to maintain a diverse population. If we don't, we tend to end up finding a kind of mediocre solution really early on and not finding anything good after that, because we kind of get trapped there. Having a diverse population will let us keep exploring the space of other solutions. And so most evolutionary algorithms focus a lot on how we can maintain diversity. And usually that comes in at the selection step. Not always, but I'm going to only talk about examples where it does. I'm not going to go into the details of any of these algorithms, but here I have 4 different evolutionary algorithms. Tournament is kind of a control, but the other 3 all maintain diversity in some way. Basically, they're trying to select things that are rarer, is the idea, mostly. Again, happy to elaborate during questions if people are curious. But the thing that's really interesting that I realized in this research is when we impose these different ways of selecting who gets to reproduce, we're actually creating ecological interactions. And we can visualize that using these species interaction networks. So this is actually the same population in all 4 of these graphs here, but with different selection methods applied to it. And so you can see they create vastly different networks of interactions. And so the big question I'm asking now in my follow-up research is, what are the evolutionary implications of those different interactions? I don't have an answer to that question yet, but I have a lot of ideas. Feel free to ask me. And so those were just examples of networks, but kind of the qualitative differences you could see between them holds up if you generate a lot of them, is basically the takeaway from this slide. Okay, perfect. Okay, so right on time, that brings me to my acknowledgement slide. So I just want to thank all the many collaborators. This work only works with lots of really great collaborators because, working on so many different topics. I also really want to thank all of the students in my lab. I've got some really great PhD students now, funding sources, and feel free to contact me via any of these mechanisms. Thank you all so much.
Raise your hand for questions.
Yeah?
What's the biggest setback in the production of the simulations and how it works or how fast they run?
Oh, yeah, okay. Well, what makes them slow, basically?
If they are.
Well, they could always be faster. So I think one of the biggest obstacles is, actually data tracking is the big one. So, if we didn't care about recording data to then analyze and understand what was happening, we could do a lot of this really efficiently. But a lot of the data that we might want to record is actually kind of slow to calculate. So for instance, a lot of the work I do involves tracking phylogenies or ancestry trees that basically represent who's the parent of who for all of evolutionary time. Those get really big. And so figuring out efficient ways to do that is actually this whole side project I have. And actually the grant I mentioned that we're working on for evolutionary epidemiology, a big goal of that is basically trying to figure out how little data we can get away with tracking so that we don't need to take the speed hit. Because the thing that we realized when we were writing that grant is we get perfect accuracy data out of these models, but we're never going to get perfect accuracy data in the real world. And so at some point, it's not worth putting a ton of energy into getting data that is so much more accurate than anything we would ever be acting on. It's almost like we're using too many significant digits or something. And so, that was a long answer to your question, but those are kind of the big things we're thinking about. Yeah?
Like you said, what kind of kit would you need to run a simulation like this?
Great question. So it depends a ton on the simulation you're running. So that simulation of antibiotic resistance, you could probably do that on your phone. That's very lightweight. Some of the work I'm doing with Matthew and Luis for the epidemiology stuff, Matthew just texted me an hour ago saying he just ran, oh gosh, what was it, a quadrillion replication events or something. That needs fancy architecture. So a big thing we're actually doing in that grant is some companies have recently been developing fancy chips to use for AI. And we've been trying to find ways to co-opt them for agent-based evolutionary modeling. And so that's what we're doing in that project. And it's working really well. But so if you want, quadrillions of replication events, you need a fancy accelerator. But a lot of these questions don't need that. And you could definitely do them on your laptop. And in fact, actually, a lot of these A-Life systems will run either on your laptop, or a lot of them have web-based interfaces. You can just go and check them out without even installing anything. It's a pretty open community. Yeah?
My brother actually had a soft robot game on his phone.
Oh, cool.
That it produces an amount of them, and then it chooses the best one, and you get to choose sort of its initial shape and see what happens.
Whoa, that's awesome. I didn't know that existed. That's so cool.
I've had it for years.
Do you remember what it was called?
Not exactly, but I should ask him. But you just choose the structures, and then you choose the muscles, and then it just has to figure out how to work those muscles to move.
Awesome. Yeah?
You made a distinction between artificial life and simulations, but a lot of their aspects seem to overlap with one another. What would you say mainly differentiates the two?
Fantastic question. I mean, you're right. It's definitely a fuzzy border. There are things that exist in like an uncanny valley between the two. It's not even an uncanny valley. It's a gradient. But I think the biggest distinction is kind of the purpose. Like a simulation is really trying to model a specific scenario in the real world, whereas an artificial life system is usually by design not trying to be exactly like the real world, other than that it has kind of qualitatively similar behaviors. Yeah?
How far out are you with like cancer research that would be able to be practically applicable in a hospital, so an oncologist could actually establish a protocol based on someone's actual genes to kill that particular cancer?
Great question. So yeah, the first clinical trial on evolutionary therapy in cancer wrapped up in 2022. It was working on kind of the simplest possible case, which is the case where there's a cost to resistance. And so in the absence of drug, sensitive cells will outcompete the resistant ones, and the resistant ones are only favored when there is drug. So this was in metastatic, castrate-resistant prostate cancer, so very specific type of cancer, definitely not like ready to generalize to everything yet. But in that clinical trial, the results were really promising. For everyone, they were able to extend dramatically the time before the cancer evolved resistance and started progressing. And for some people in that clinical trial, it actually still hasn't started progressing. They've been able to kind of just maintain it as like a long-term chronic problem. And for the people who did eventually evolve resistance, it actually looks like the problem was just that they gave them too much drug. They kind of didn't do evolutionary therapy hard enough. So yeah, that's been really encouraging. But that is like the best case for using evolutionary therapy on cancer. The kind of thing you were talking about with like identifying specific genes and stuff is what would be necessary when the trade-offs between being resistant and not being resistant are more complex. And so on that, we're definitely still a ways out. So yeah, Jake's lab at Cleveland Clinic, like he's an oncologist. He like, works one day a week in a clinic. He has patients. So that's like, climate change is what keeps me up at night. That's what keeps him up at night. And yeah, he's always like, thinking about how to get things into the clinic as fast as possible. And like, I think we're making gradual progress. So like a big thing that they've been working on and we've been collaborating on them with is this kind of game theory lens for figuring out when is there a situation where it would be easy to use evolutionary therapy. And so I could see like a slightly more sophisticated clinical trial starting up soon. But yeah, I mean, it's still a long road, especially because like this feels scary to like a classically trained oncologist, right? We're saying, hey, maybe you should give the patient less drug. It makes sense from an evolutionary perspective, but if you're used to thinking, oh yeah, drugs kill cancer, give all the drugs, it sounds like kind of a wild thing to do. Yeah?
How far about the evolution, the climate issues with growing and stuff? Like what kind of research and stuff are they doing on that?
Great question.
I personally had issues with my garden this year, so.
Sorry to hear that. So I'm actually okay, my goal is in maybe 5 to 10 years that I'll pick up that research direction. So I haven't started thinking a ton about the practicalities yet. I've been keeping my eye on what people are doing. There's definitely a lot of work on soil microbiomes and trying to figure out what's important in them. There's been work really by the ag industry on kind of drought tolerant plants and stuff like that. I don't know if I've seen kind of a holistic how do we construct an ecosystem that will keep crops alive yet, because I mean the reason I'm not working on it yet is I don't think we're ready to work on it. I don't think we know enough about the basic ecology and evolution. So yeah, I guess I don't have a great answer to your question. I mean, it's definitely a thing people are thinking about. It's a reality that we're facing.
I don't know. I kind of disagree. I think we do know enough. It's just the industrial farming isn't ready to adapt to it, so it's a political thing.
That's absolutely a component.
There is tons of information out there about how to make your own yard microbiome sufficient, especially because I've done a lot of research on this. Gardening is my thing. but I had to go from ground to containers,
Mhm.
and that's a whole, so I had to learn how to bring that microbiome to my container.
Oh, that's fascinating.
So it's been great. I mean, everyone suffered. My tomatoes, I still had tomatoes until the very first frost we had in the end of October. And I mean, it was just amazing. And I'm like, evidently, I did something right.
Oh, that's fantastic. That's great to hear. Yeah, no, and I also think there's a lot of like, you know, things we can do in the present, you know, like shifting to containers, you know, like shifting what crops we grow based on, you know, the new environment that we're in. Totally lots of like kind of stopgaps like that. I don't know if like there's a point where those break down eventually or not. That's kind of like the time horizon I'm thinking about.
That's where the study really isn't, because there hasn't been a study on long-term
Exactly.
viability of it. But I do know that, or I believe we do have the information to where we can get started. But like you said, you don't think enough people are ready for it, which I don't think they are.
It's really good to know that that the research is starting to be there, though. That's great.
Yeah, well, it's all thanks to Michigan State University, honestly, because they're the last ones that I read the article of, especially when Oregon started going after the farmers.
Mm.
And so someone did a wonderful paper on is farming gardening industry going down, and it was a great article because it talked about the microbiomes and everything.
Oh, cool. That sounds fascinating. Yeah?
How do you avoid over-influencing your results like when you're doing this? Because I know it's all computer-based, and you can probably a fine line, I'm assuming, to kind of skew it one way or the other.
Yeah, it's a really good question. So for me, what I try and do is I try and stay really focused on like what my actual question is. Because I think that the biggest place that can come in is like kind of your question can shift. Like there's a difference between, you know, it being likely that something will happen and it being possible to make that thing happen if you come up with a contrived enough circumstance. So, you know, a lot of the time what I try and do is frame my questions around like, okay, so there's a lot of, you know, variables that are going to affect whether some evolutionary outcome happens. And so instead of asking, does that thing ever happen, I might ask, like, what is the range of variable values? Like, well, what is the part of parameter space where that thing happens? How large is that space? Is that space something that happens in real life or not? So I think that's a helpful tool. I mean, the other thing is actually, I think, a big strength of these systems. So it's really easy to write like a mathematical model that like forces something to happen. A thing that's nice about having these kind of messier systems is we can pull back a lot of the assumptions that we make when we make mathematical models. You can say, okay, so you know, we started out by like hard coding that this thing interacts with this other thing in exactly this way, and okay, sure, we got the result we expected. But now let's take a step back. Let's, you know, let the way that that thing works evolve. Let's see what happens then. Okay, now let's let it evolve via something that's closer to how DNA works. And like we gradually add in more of the messy real world complexity and see if our result holds up in light of that. Mm-hmm.
What were you studying that gave you the most surprising, unexpected result?
Oh, that is such a good question. Oh, I feel like, okay, that question deserves a really good answer. I'm like totally not thinking of one right now.
Because you're kind of like dabbling in everything
Right.
now when it comes to it. So like I'm just, especially when you do artificial life, like I mean, is there something that really sticks out that it predicted that you were like, I never would have even thought about that, you know? I mean, because with computer programming, I mean, it can give you the world.
Totally, yeah.
So I'm just wondering, like, what was the most shocking thing? Like, oh my gosh, I never would have thought about that.
Okay, so all the examples I'm thinking of are gonna like sound really boring without having like the context. You know, for instance, you know, I study these ancestry trees as a way of like recording the evolutionary process. There's a whole horde of assumptions that people who've been studying those for years have made about how they tend to look. And the ones we get out of these artificial life systems look nothing like that. And it's for reasons that make sense now that I thought about it, which is true of a lot of evolution. Like first you see it and you're really surprised, and then you think about it more and it makes sense. But yeah, that was actually pretty surprising. I guess the other thing actually, so the ecological networks in evolutionary computation that I was just talking about, so I didn't talk about the math side of that project because it's kind of dry, but it turns out that there were two people in the 80s that actually, one was in evolutionary computation and one was a theoretical ecologist. They came up with the same equation, like within a year of each other, and like they didn't notice. And so like, one of those algorithms for selecting who gets to reproduce is actually mathematically equivalent to models that have been used in ecology forever.
It's amazing how things just connect.
Yeah. Probably the most surprising result that I know of someone getting though, and this is just such a fun story that I love to tell it. So my PhD advisor, Charles Ofria, who developed Avita in the first place, he was trying to do an experiment where he didn't allow beneficial mutations for reasons I'm happy to talk about. But basically, yeah. So every time a new organism was born, before he let it go into the population, he was testing it to see if it was reproducing itself faster than its parent. And if it was, he wasn't letting it into the population because he only wanted deleterious mutations that made fitness worse. And so he did that, and at first fitness wasn't going up. Great, that's what you'd expect. But then it started going up, and he was like, "What is happening? How is it going up?" And so he looked. And it turned out in the test environment where he was testing to see if there was a beneficial mutation, he was using a specific set of input numbers that they could take in from the environment. And they had learned to recognize those numbers, and they were playing dead in the test environment so that they could get into the population.
Oh, so it evolved.
Yeah.
That's amazing. That's kind of scary though, too.
Yeah. Yeah?
Your lab does a lot of different things, and I'm wondering kind of why you do so many different things. Is it because they're all interconnected, or is it because you do a bunch of things, you hope one of them works? Or is it that if you focused on one, things take time, and so you can do one for a while and work on another one? Or why?
Yeah, I'd say it's because they're all interconnected. So I think, I'm trying to pare down slightly the number of things my lab does because it's getting a little out of control. But every time we accidentally spawn off a new project that feels like it's in a new field, it's because something else we did gave us the idea for it. So I have a student that just did a swarm robotics project. That's kind of an evolutionary computation, but it's also kind of its own thing. But the reason he did that is that in the origin of life stuff, we learned something about what makes groups more likely to evolve to work together. And we thought, oh, that would help swarm robotics. We should try it out. And so it's pretty much all driven by ideas. Some of it is also driven by funding, obviously, and driven by collaborations. But a lot of it is just we see an idea in one place and we think, oh, you know what field needs that idea? It's this other one. Yeah?
What's your favorite thing that you guys are working on in the lab right now?
Oh gosh, I feel like it's really hard to choose a favorite. It changes based on whichever student I've talked to the most recently. Okay, this is, I don't know if this is actually my favorite, but one thing that's really cool is, so I have a student who is really interested in, taking a step back. So there's been some research on deep neural networks and other kind of machine learning image recognition systems that, you give them an image, they say, oh, this is a picture of a dog or whatever. There was some research a couple of years ago that shows that you could fool those pretty easily. If you try to evolve an image that they will say is a dog, you can evolve an image that looks nothing like a dog that they will say is a dog. It could be an image of static and they'll still be confident it's a dog, which has, legitimate security concerns as we use these systems more and more. So I had this one student who was actually just kind of messing around with a class project and found that there are some machine learning representations that basically seem harder to fool in that way than others. And so now we're doing basically a study on why is that? Which ones are easier to fool and why, and can we use that? Specifically, he's found this one kind called support vector machines aren't very foolable so far. So maybe we should be using them in more places where we're worried about this security risk. yeah, that's an early project. We haven't written it up yet, but I think that's something I feel like could have like really important implications. And so, yeah, it's the one I'm most excited about right now, but it'll change tomorrow. This is why I keep accumulating so many projects. They're all exciting. Cool. Any other questions? Oh, yeah?
When finding antibiotics for bacteria, does it ever feel like a game of tag where you find an antibiotic and that bacteria over some time finds some way to be immune to it? And if so, how long does it take for it to find immunity?
Great question. I mean, so fortunately, I'm not in the business of finding antibiotics, so I don't have that feeling of playing tag, but people who do definitely are. Same with people who develop cancer drugs. And the answer is, it depends a little bit. Some, like we have a few kind of like, last resort antibiotics that there isn't much resistance to yet, and we're specifically, avoiding using them too much so that we don't put too much resistance to them out in the environment. So evolution of resistance in bacteria is complicated because unlike in cancer, they can share genes with each other pretty easily. So once resistance is out there, like it's often pretty easy for it to get spread around. And so it's often less a matter of getting a long sequence of mutations to get the resistance, and more a matter of like finding a friend that already has the resistance to get it from. Obviously, it's not intentional, but that is a thing that happens. Yes, yeah, that is a problem. It's frustrating, but that's why we're thinking about ways to resensitize populations, ways to channel the ways they evolve resistance so that we can maybe kill them with a different drug afterward. The more drugs we have, the more tools we have to try and push the population around. Although also not just drugs. Like, so, I mean, the idea I was just kind of, making casual reference to is called collateral sensitivity, where evolving resistance to one drug makes you sensitive to another drug. And people have mostly studied this with drugs, but you can actually do it with other things too. Like, there's this paper I love by Jeff Maltus and Kevin Wood, where they did the same experiments we usually do with drugs, but just with things from their spice cabinet. Like, they looked at how evolving, in the presence of cinnamon or something affects the sensitivity of the bacteria to various drugs in the long run. And so, we think about drugs as being our only kind of levers for pushing the population around this evolutionary space, but they're not actually. There's other things we can use too. And I think, the more we get creative, the more we might be able to really make progress. Mm-hmm.
Like with COVID and the vaccine that came, or vaccines, there was some criticism that the testing of the vaccines was done too quickly. But I assume with a lot of the work you're doing, the testing could be accelerated with the computer simulation modeling. Is that going to make it easier for things like that, or new things that come up?
So you have to be really careful, because it's really easy to have something in your simulation that is just different than the way humans work. Human biology is complicated. Human immune systems in particular are very complicated. I'm terrified of trying to model immune systems. So, I mean, yes, the more we can kind of do preliminary pilot experiments digitally, the more we can, go into actual human trials with a pretty good idea that something's going to work. But it's still, you're always going to need the human trials, because you can't be sure from hamster trials that it's going to work in humans. You can't be sure from a computer that's going to work in humans either. I mean, maybe if we get some hyper realistic, very well validated model of the human immune system one day, maybe we could use that as a really good starting point. But I still have a hard time imagining we could ever just totally trust it and apply something at a population scale before testing at a smaller scale in humans.
Well, thank you.
Thank you all for great questions.